In this video, we're going to talk about what is an enzyme, and we're going to learn how we classify different enzymes depending on the type of reactions that they catalyze. So an enzyme is a protein. It's a class of proteins that participates in chemical reactions. And what they specifically do is they behave as biological catalysts, meaning they are able to speed up the rate of a chemical reaction, and it is not consumed in the process. So what this means is that once it participates in speeding up a reaction, it can go ahead and then speed up another reaction since it wasn't consumed. So here's an image of an enzyme, and I'm showing you some keywords here that we use to talk about the enzyme and how it participates in a reaction. So the substrate here is going to be one of the reactants in the chemical reaction that it is speeding up. So this substrate is going to be part of the reactants, and then these reactants are going to bind to your enzyme shown here in blue, and they're going to specifically bind to the active site. So the active site on an enzyme is the region on your protein that is going to recognize the reactant in your reaction. And that is what is being shown here. So instead of saying reactant, I will call this a substrate because that is how we refer to a reactant when it is part of an enzyme complex. So here I have an enzyme substrate complex. What this means is I have an enzyme, and to it, the substrate is bound in the active site. So the enzyme will go ahead and bind the reactant in the active site, and then the chemical reaction will proceed so that then the products are released from the enzyme, and the enzyme can then be reused again to bind another substrate or another reactant. It'll bind another substrate and then carry out the reaction again. So notice how the products here, basically what this cartoon enzyme did here is it broke the bond between the green spheres and the red spheres, where initially when they were substrates, they were linked together, then they bound the active site, and then they were able to be released individually. So this means there was some sort of bond cleavage in this reaction in which the enzyme participated. Enzymes have a few other features that are important for their function. So enzymes are said to be very specific, or that they show high specificity. What this means is that the enzyme can recognize either a specific substrate, so it can be very specific to just a single reaction, meaning there's only a particular substrate that it will recognize and bind to the active site, or enzymes can catalyze a particular type of reaction, meaning the substrates can look a little bit different from each other, but they will always carry out the same peptide bond cleavage, for example. Okay, so that would be an enzyme participating in a class of reactions. Okay, so enzymes are very specific, and this is important for function because you don't want the enzymes in your body to just be carrying out any reaction that they want. You want them to carry out the specific reactions for which that enzyme is needed, okay, because all the reactions in your body need to happen at certain time. Enzymes can often require a cofactor as well. Cofactor is defined as a metal ion. So a cofactor can be a metal ion or a non-protein organic molecule, such as vitamins, for example. And these are considered cofactors when they are needed for an enzyme catalyzed reaction to occur. So what this means is that metal ions, which we consume in our diet, this is why we need little ions like magnesium and sodium and so on in our diet in order to be able to supply those metal ions in these type of enzyme catalyzed reactions. And enzyme catalyzed reactions are going to be pretty much everything that is involved in metabolism. So we use vitamins and metal ions in order as cofactors in order to be able to give the enzyme its function. So in other words, the enzyme cannot catalyze a reaction if it doesn't have these cofactors. Not all enzymes need cofactors, but many of the coenzymes in metabolism, as we will see in the next unit, they will require cofactors. Lastly, the enzyme names, you're going to see that they generally end in ASE or ACE. So oftentimes, just from the name alone, you can tell whether you're dealing with an enzyme.